Okay, let's talk about cell division now. What I have here is several phases of mitotic cell division. Mitosis is normal somatic body cell division. And we have to continue from where we left in the cell cycle. You might remember there is a G2 phase, an interphase. Interphase is like, the period where the cell is not dividing is called interphase. And in the G2 period of the interphase, the cells get ready for a division. What happens in the interphase, G2 phase? The chromatin is quite nicely defined. The nucleus is clear. Um, the centrosomes show two centriole pairs here, which are close to each other. The cell is just getting ready, preparatory phase. Sorry, interphase is just no cell division phase. Then comes prophase, P. Pro is the first, right? So before. So in the prophase, the cell prepares for the actual cell division. How does it prepare? The centrosomes, the centrioles of the centrosomes, they move far apart from each other. They start showing early mitotic spindles and some asters like rays. The chromatin becomes more clearly defined. Now you will see chromosomes showing replicated materials, chromatids, so sister chromatids are visible in prophase. And this is when synapses, which is crossing over, also happens. The kissing and the exchange of genetic materials, the recombination happens in prophase. Then comes the migration phase. It's called metaphase. I call it migration phase. So you remember what happens. The two centrioles now move to the opposite poles, north and south pole of the cell. And all of these chromatids, chromosomes, they line themselves in the equator. So the equatorial alignment and migration happens in metaphase. In anaphase, now you get allocation of materials to two sides of the cell. That's anaphase. Anaphase is allocation. How does the allocation work? There's equatorial divide of the nuclear material. The daughter chromosomes are born. The material gets allocated to the two daughter chromosomes. And then finally, the cells tear apart in the telophase. So you see a bend forming in the membrane, a cleavage forming, and each part becomes its own, and the cells tear apart. This is telophase. Telo is the end, pro is the beginning, and meta and ana are the middle two phases. So this is a representation of what happens with genetic material during a mitotic cell division. What is mitosis? Mitosis is the normal growth-related bodily cell division where you don't lose any genetic material. So you start with a diploid chromosome, which is 2n, 46 for us. I cannot draw all the 46, so I've just 4 of them here. So 2n is the number you start with. And when you're ready in the prophase phase, you get chromatids. You still have no change in the number of chromosomes, and the number is 2n. You might say the material has replicated, so it becomes 4n, but the actual fact is, if you look at the count, it remains as 2n, okay? And then what happens? The cells divide into two. Again, they all have same number of chromosomes. So there is no change in the number of chromosomes. There is a brief period when nuclear material is doubled, but that's only brief period. And the cells actually divide. The daughter cells are same amount of chromosomes, 2n. So this is called a preserved cell division where there is no reduction in chromosomal numbers. But meiosis is a different story. Meiosis takes place in germ cells, and meiosis is a reduction cell division. Why do we call it a reduction cell division? Because you start with a 2n diploid cell and you end up getting daughter cells which have only 1n, half of the chromosome, haploid. So how does that happen? Meiosis must be thought of as a complicated cell division which has two phases. The first phase is meiosis 1, which is very similar to, so meiosis 1, it's not similar to mitosis. Meiosis 2 is similar to mitosis. But meiosis 1 is where the reduction happens. So the 2n becomes 1n and 1n. And then in meiosis 2, you have mitosis-like cell division. In meiosis 2, mitosis-like cell division, where 1n becomes 1n and 1n. So this is preserved cell division, but this is reduction cell division. So because there are two divisions happening, the material gets halved. One of the divisions is a halving division. The other one is a preserved division. So meiosis should be thought of as a cell cycle, a cell division process that incorporates two division points. One is a mitosis-like division point that happens later, but the first one is a reduction division. And this takes place in germ cells.